Hello students, welcome to EPG Pathshala. I am Ramesh Kothari, Professor of Microbiology, UGC CS Department of Biosciences, Saurashtra University, Rajkot, Gujarat. Welcome you all. Today we are going to discuss about glycogenesis from the paper Metabolism of Carbohydrate. Glycogenesis, a process of glycogen synthesis. Glycogen is a highly branched, very large polymer of glucose linked along its main line by alpha 1,4 glycosidic linkages branched arise by alpha 1,6 glycosidic bonds about every 10th residue. Glycogen occurs in cytosol as granules, which also contain the enzymes that catalyze its formation and utilization. Glycogen is the storage form of glucose. Its polymeric nature allow the sequestering of energy stores with much less of a problem from osmotic effects than glucose would cause. Now let's see the objective of this lecture. In this lecture, we will discuss how glycogen is synthesized, what is the role of glycogenin in glycogen synthesis, the process of branching in glycogen, and underlying biochemistry of glycogen metabolism. This diagram shows the concept map that how glucose stored in a form of glycogen. Looking at figure, it is clear that the formation of glycogen, starch and sucrose, which are the various form of storage of carbon, are formed from the glucose, and this figure also shows that how the glucose is converted into glucose, starch and various other source of carbon in different ways. As mentioned earlier, glycogen is a highly branched large polymer of glucose molecule linked along its main line by alpha 1,4 glycosidic linkages. Branches arise by alpha 1,6 glycosidic bond at about every 10th residue. The above figure shows the structure of glycogen. Looking at structure, it is clear that the formation of long chain polymer of glucose is achieved by the formation of alpha 1,4 linkage between two subsequent glucose molecules, and this two non-reducing end of the glucose polymer chain, when joined together through an alpha 1,6 linkage, it form a branching point there. So in the structure of glycogen, there are two ends. One is reducing end and other are non-reducing ends. Thus, it gives a very complex and highly branched large polymer of glucose molecule. Glycogen occurs in the cytoplasm as granules. Granules also contain the enzymes and regulatory proteins which is required for its synthesis and degradation. Glycogen, which acts as an important energy reserve for the body, it is stored in liver and skeletal muscles. Glycogen stored in the muscles utilized for energy requirement of muscles only, while glycogen stored in liver utilized for energy requirement of the rest of the body. Glycogenesis is the process of glycogen synthesis in which glucose molecules are added to chain of glycogen for storage. This process is activated during rest period following the Cori cycle in the liver and also activated by insulin in response to high glucose level. Glycogenesis is the biosynthesis of glycogen, the major storage form of carbohydrate in animal similar to starch in plant. Glycogen is used to rescue when the blood glucose drops down, a situation which prevail between our daily meal. The major site of storage of glycogen are liver and muscles. Although the glycogen content of liver is greater than that of muscle, 3/4 of total glycogen is stored in muscles due to their mass. This figure shows an overview of glycogenesis. It explains the formation of glycogen from glucose. In the synthesis of glycogen, one ATP is required for every glucose unit incorporated into the polymeric branched structure of glycogen. The glucose in the form of glucose 6-phosphate is synthesized directly from glucose or as the end product of gluconeogenesis. The enzyme hexokinase phosphoglucomutase UDP-Glucose Pyrophosphorylase and Glycogen Synthase play an important role in the process of glycogen synthesis. New glucose residue is attached to primer by glycogen synthase. Glycogen synthase catalyzes the transfer of glucose from UDP-glucose. New glucose molecule transfer to the C4-hydroxyl group at the non-reducing end of growing glycogen molecule. Looking at the overview of glycogenesis, we can see that the glucose is converted into a glucose 6-phosphate by the enzyme hexokinase, and ATP is converted into ADP. This reaction is non-reversible. Now, the glucose 6-phosphate is reversibly converted into glucose 1-phosphate by the enzyme phosphoglucomutase. There is no either input or production of energy during this reaction. Now, glucose 1-phosphate is irreversibly converted into UDP-glucose. UTP is converted into UDP, and this UDP is combined with glucose, releasing pyrophosphate molecule, and this reaction is catalyzed by UDP-glucose pyrophosphorylase enzyme. Now, this UDP-glucose is used for the synthesis of glycogen, and it's at the end of addition of many molecules of UDP-glucose, it leads to the formation of glycogen. So, students, as we all know that muscles and liver are the major sites for the storage of glycogen, and although its concentration in the liver is higher, the much greater mass of skeletal muscles store a greater total amount of glycogen. Liver can mobilize its glycogen for the release of glucose to the rest of the body, but muscles can only utilize its glycogen for its own energy needs. The glycogen stores allow us to eat intermittently by providing an immediate source of blood glucose for use as a metabolic fuel. In human, the liver glycogen stores are only adequate for about 12 hours or less without support from the gluconeogenic pathways. There are various steps of glycogenesis. Glycogen synthesis require three major enzymes and occurs by a separate pathway from glycogenolysis. The first step is UDP-glucose formation by UDP-glucose pyrophosphorylase. The second step is glycogen synthesis by glycogen synthase, and the third step is glycogen branching. So these steps successively result in the synthesis of a glycogen starting from UDP-glucose formation to glycogen synthase to glycogen branching. As shown in the figure, glucose is activated before it gets polymerized into glycogen. Glucose is phosphorylated to a glucose 6-phosphate by hexokinase enzyme in muscles or glucokinase enzymes in liver. For further anabolic polymerization reaction, it is converted into glucose 1-phosphate by the reversible action of phosphoglucomutase. Glucose 1-phosphate is then attached to UTP by the action of UDP-glucose pyrophosphorylase from UDP-glucose and pyrophosphate. The carbon 1 of the glucosyl unit being esterified to the diphosphate moiety of UDP. The standard free energy of this reaction is very less, that is nearly zero. But the pyrophosphate formed is hydrolyzed to orthophosphate in a high exergonic reaction, Conversion of pyrophosphate to orthophosphate is catalyzed by inorganic phosphatase. Uh, Looking at the reaction, glucose uh, is converted to UDP-Glucose through three reactions catalyzed by hexokinase, phosphoglucomutase, and UDP-Glucose pyrophosphorylase successively. And at first step, the ATP is converted into ATP, and in last step, the UTP is converted into UDP, releasing two pyrophosphate molecules. This slide shows the structure of UDP glucose. The glucose combined to uridyl diphosphate. UDP glucose is the activated form of glucose used for metabolism of galactose and biosynthesis. UDP glucose is a high energy compound that can donate glucosyl units to the growing glycogen chain. No further energy is required for glycogen synthesis. Now, in step 2, the glycogen synthase transfer the glucosyl residue from UDP glucose to the non-reducing terminal residue of glycogen. It is transferred to hydroxyl terminal of C4 terminus of glycogen to form an alpha 1 4 glycosidic bond. This reaction is catalyzed by glycogen synthase. It is the key regulatory enzyme in glycogen synthesis. Glycogen synthesis require a primer. Glycogen synthase can add glucosyl residue only if the polysaccharide chain already contains more than 4 residue. It means that glycogen synthase can only extend an existing chain. If there is no primer, chain cannot grow. Priming function is carried out by glycogenin. You can see this reaction in the figure that UDP glucose, when combined with glycogen, n number of glycogen residue, uh, which results in the subsequent release of UDP molecules and growing glycogen chain. So, glycogen synthesis by glycogen synthase and glycogenin. De novo synthesis of glycogen occurs since glycogen synthase can add a new glucose molecule to existing chain. Glycogenin is a primer for glycogen synthesis. It is a protein composed of two identical subunits. It is a 37 kilodalton protein that is glycosylated on a specific tyrosine residue. Glycogenin contains 8 glucose unit linked by alpha 1,4 linkages. These glucose molecules are added to the protein by autocatalysis. This figure shows the formation of glycogenin primer. The first glycogen molecule is attached to hydroxyl group on tyrosine, which is located on 194th position of glycogenin, which is catalyzed by the subunit of glycogenin. Then it is autocatalytically extends the glucan chain up to 7 residue long. This glucose molecule is donated by UDP-G. In this form, glycogenin can act as a primer. If you see in the figure, initially glycogenin, when combined with tyrosine, and this complex glycogenin tyrosine complex is again combined with glucose, and hence glycogenin tyrosine glucose complex is formed. Now this undergo autocatalysis, and really hence the addition of this UDP glucose molecules results in the growing chain of glycogen. This figure shows step 3, that how branching of glycogen occurs. Glycogen synthase catalyzes only alpha 1,4 glycosidic bonds, which results into the formation of alpha amylose. Branching is catalyzed by separate enzyme called branching enzyme. It is also known as amylo 1,4 to 16 transglycosylase. After a number of glucose units have been linked as a straight chain with alpha 1,4 linkage, branching enzyme break alpha 1,4 bond. It breaks a 7-unit segment of alpha 1,4 residue from a glycogen chain and transfer to a C6 hydroxyl group, that is hydroxyl groups present on carbon 6 of a glycosyl residue, that is 4 residue away from an existing branch. Reattachment is done by creating an alpha 1,6 bond. Branches are very important in a growing chain of glycogen. Enzymes involved in glycogen synthesis, that is glycogen synthase, and glycogen degradation, that is glycogen phosphorylase, act only at the end of the glycogen molecule. More terminal ends increase the rate of synthesis and degradation of glycogen. From figure, you can see the growing chain of glycogen and branching enzymes which result in the multiple branching of glycogen. So, as we discuss that glycogen is an important storage food for cell. In conclusion, we can say that glycogen is found in the form of granules in the cytosol in many cell type and play an important role in the glucose cycle. It functions as a secondary short-term energy storage in animal cell also. Glycogen forms an energy reserve that can be quickly mobilized to meet a sudden need for glucose. We know that cell require source of energy at any stage. We also know that cell is not under constant condition all the time. When cell is growing under luxurious nutrient conditions, it store energy. As and when cell require this energy, this type of storage source of energy can be mobilized, which convert from glycogen to glucose, and then glucose becomes available in glycolytic pathways. It catabolizes under amphibolic conditions, sometimes under aerobic conditions, sometimes under microaerophilic conditions, and sometimes under anaerobic conditions. And when it catabolizes under aerobic conditions, we know that from glucose to it convert to pyruvate, and from pyruvate to acetyl CoA by an enzyme system. And once acetyl CoA is synthesized, it enters in the TCA cycle to provide a plenty amount of energy through various mechanisms. So this is all about glycogen synthesis. Thank you.